Welcome, learners, to another deep dive. Today, we're taking a plunge into one of the most fundamental yet intricate processes in your body. How oxygen and carbon dioxide are transported through your blood.
Right. And this is really crucial stuff for anyone studying human physiology, medicine, or frankly, anyone just curious about, the breath of life.
We're drawing our insights today from a foundational text, Medical Physiology, by Boron and Boulpaep.
And our mission really is to unpack these complex concepts, sort of from the ground up, make them accessible, help you build a clear mental picture without needing diagrams in front of you.
And we'll connect the dots to real-world clinical scenarios so you can see why this knowledge is so vital.
Exactly. Why it matters for diagnostics, for treatment.
So let's unpack this journey, starting with the vital gas we absolutely cannot live without. Oxygen.
Yeah.
Now, you might think blood just dissolves oxygen,
Right.
like sugar dissolving in water. But if you look at the actual numbers, you pretty quickly see why that's not nearly enough.
Yeah, imagine trying to fuel your entire body's metabolism with just dissolved oxygen. The amount is incredibly small, only about 0.0003 milliliter of O2 per deciliter of blood for every millimeter of mercury of oxygen partial pressure. Really tiny.
Right. So if our tissues tried to extract all of that dissolved oxygen and our heart was pumping at its normal rate, say 5,000 milliliter per minute, we'd only deliver about 15 milliliter of O2 per minute.
Which sounds like maybe a lot, but the average resting human consumes around 250 milliliters of oxygen per minute.
Wow.
So to meet that demand with only dissolved oxygen, your heart would literally have to beat nearly 17 times faster. It's not feasible.
Clearly not.
No. And this highlights the absolute necessity of our next player, hemoglobin.
Okay, so over 98% of the oxygen carried in your blood hitches a ride on hemoglobin, specifically inside your red blood cells. What exactly is this amazing molecule?
Well, hemoglobin, or HB, is a large, pretty complex protein. Think of it as having four parts. It's a tetramer, meaning four subunits.
Okay.
And each subunit has two main components: an iron-containing heme group, that's where the action happens, and a polypeptide globin chain.
Right.
In adult hemoglobin, we have two alpha globin chains and two beta globin chains. So 4 chains, 4 heme groups.
And where does the oxygen actually attach to this structure?
So each of those 4 heme groups contains a single iron atom. And that iron atom is the direct binding site for one oxygen molecule.
Got it.
And it's really crucial that this iron is in a specific state, the F2++, or ferrous, state for oxygen to bind.
F2 plus A. Okay.
Now, sometimes things can go slightly wrong. That F2+ can get oxidized, meaning it loses an electron and becomes F3+ ion.
Right.
When that happens, it forms methemoglobin, or MedHB. And the problem with MedHB is that it can't bind oxygen.
It's basically useless for carrying oxygen then.
Exactly. It reduces the blood's overall oxygen carrying capacity.
So if that oxidation happens, you've got this MedHB floating around.
Yep.
Is there anything our body does to fix that, or are we just stuck?
That's a great question. Our bodies are pretty clever. Inside the red blood cells, there's an enzyme called methemoglobin reductase.
Okay.
Its job is specifically to convert that non-functional MetHB back into functional hemoglobin, back to the Fe2+ state.
So it reverses the oxidation.
Precisely. But if there's a genetic defect in this enzyme, MetHB levels can rise significantly, and that can lead to tissue hypoxia, lack of oxygen in the tissues.
Ah, okay.
It's a really critical example of how a single molecular detail, like one enzyme's function, can impact the whole body's oxygen delivery. It shows why these systems are so finely tuned.
Okay, so this binding of oxygen to iron, it needs to be reversible, right? It can't be like rust, a permanent bond. How does hemoglobin manage that delicate balance?
Right, good point. The globin portion is absolutely crucial here. It doesn't just hold the heme, it actually cradles it in a very specific way.
How so?
Well, this allows oxygen to bind loosely, reversibly. And there's a key amino acid, histidine, that directly binds to the A2+, and it helps stabilize that temporary oxygen-iron complex without making it permanent.
So the globin chain sort of protects the interaction.
In a way, yes.
Right.
And this is where cooperativity comes in, which is what makes hemoglobin so special.
Cooperativity, what does that mean exactly?
Think of hemoglobin as, maybe like a group of 4 friends deciding whether to open a door. When no oxygen is bound, the iron atom is pulled slightly out of the flat plane of the heme ring, and it tugs on that histidine.
Okay.
This creates structural strain, putting the whole molecule in a tensed or T state. And this T state has a very low affinity for oxygen. The door is hard to push open.
So it's like hemoglobin is kind of closed for business to oxygen initially.
That's a good way to put it. But when one oxygen molecule does manage to bind, maybe with a bit of effort,
Yeah.
it pulls that iron atom back into the plane of the heme ring. This relieves the strain, and that small change gets transmitted across the entire hemoglobin molecule.
Yeah.
It causes all four subunits to sort of snap into a different shape, a relaxed or R state.
Ah. So one oxygen binding changes the whole molecule's shape.
Exactly. And this R state, it loves oxygen.
So the R state has a much higher affinity.
Astonishingly higher, about 150 times higher affinity for oxygen than the T state.
Wow, 150 times.
Yeah, and this explains the characteristic S-shaped or sigmoidal oxygen hemoglobin dissociation curve we see.
Right, the curve isn't linear.
No, at low oxygen levels, like in the tissues, it's hard to get that first oxygen to bind. Low affinity T state. But once one binds, bam, the affinity shoots up, making it much easier for the second, third, and fourth oxygens to bind quickly, high affinity R state.
So it helps load up oxygen efficiently in the lungs where there's lots of O2.
Precisely. And then, importantly, when it gets to the tissues where oxygen is lower, the reverse happens. As one O2 comes off, it encourages the others to leave too. It's this cooperative binding and release that's foundational to efficient oxygen transport.
That S-curve really paints a picture. But hemoglobin isn't the only oxygen-binding protein we have, is it? What about myoglobin? Where does that fit in?
Right, good question. Myoglobin is structurally similar, kind of like a single subunit of hemoglobin, but you find it specifically in muscle cells.
Okay, just in muscles.
Yeah, it binds only one oxygen molecule, not four. But crucially, it has a much higher oxygen affinity than hemoglobin does, even than hemoglobin in the R state.
Higher affinity, why is that important in muscle?
Well, think of it as helping to pull oxygen out of the capillaries and speeding up its diffusion through the muscle cell to the mitochondria, which are the powerhouses constantly using oxygen.
So it acts like a little shuttle service inside the muscle cell.
Exactly. Or maybe like a local, rapidly accessible storage tank. It helps maintain a steep oxygen gradient from the blood into the mitochondria, ensuring they get the O2 they need, especially during exercise.
Okay, let's go back to that hemoglobin dissociation curve. You said it plots how much oxygen hemoglobin holds at different oxygen partial pressures, PO2.
That S shape is the key takeaway. So at the high PO2 we find in arterial blood, maybe around 100 millimeters of mercury.
Right, in the lungs.
Yeah. After loading up in the lungs, hemoglobin is nearly saturated, about 97.5% full of oxygen.
Okay.
But then, as that blood travels to your systemic tissues, your muscles, your organs, the PO2 drops. In mixed venous blood, it might be around 40 millimeter Hg.
Much lower.
Much lower. And on that steep part of the S-curve, this drop in PO2 causes hemoglobin saturation to fall significantly, down to about 75%.
So what does that drop from roughly 97.5% down to 75% actually mean for oxygen delivery?
That difference, that 22.5% drop in saturation, represents the oxygen that hemoglobin releases or unloads to the tissues. Per deciliter of blood, that translates to about 4.5 milliliters of O2. And that amount accounts for nearly 96% of all the oxygen delivered by your red blood cells.
So almost all the oxygen delivered comes from hemoglobin, not the tiny dissolved amount.
Exactly. We basically spend our lives cycling between those 2 points on the curve, loading up near 100% in the lungs, dropping down to 75% in resting tissues, more in exercising tissues, and delivering that vital difference.
Okay, that makes sense. So what happens if our hemoglobin levels themselves aren't right, like in anemia?
Right. If you have anemia, meaning you have decreased hemoglobin concentration in your blood, your total oxygen carrying capacity is reduced. Even if the hemoglobin you do have gets fully saturated in the lungs, there's just less of it to carry the oxygen.
So the blood carries less oxygen overall.
Yes. The body tries to compensate, maybe by increasing cardiac output, pumping blood faster, or by extracting a larger percentage of oxygen from the blood that passes through the tissues.
That can lead to symptoms, right?
Absolutely. Things like pallor, looking pale, lethargy, fatigue, maybe palpitations, shortness of breath. All signs that the tissues aren't getting enough O2 consistently.
And the opposite condition, polycythemia. Too much hemoglobin.
Yeah, that's not good either. While it increases the oxygen content of the blood, having too many red blood cells makes the blood much thicker, more viscous.
Like sludge.
Kind of. This increased viscosity makes it harder for the heart to pump the blood. It can lead to high blood pressure and can actually impair gas exchange in the lungs sometimes. There's definitely an optimal range for hemoglobin and hematocrit.
And that classic blue skin we sometimes see in patients, Cyanosis. That's related to hemoglobin too.
Yes, directly. Cyanosis is the visible sign, that bluish tint to the skin and mucous membranes caused by the presence of a certain amount of desaturated, purplish-colored hemoglobin in the capillaries.
So it's about the deoxygenated HB, not the lack of oxygenated HB.
Exactly. Which leads to an interesting point. An anemic patient might have dangerously low oxygen levels, but they might not look cyanotic because they simply don't have enough total hemoglobin, saturated or unsaturated, circulating for that absolute amount of the oxygenated hemoglobin needed to cause the visible blue color to be reached. So absence of cyanosis doesn't always mean good oxygenation, especially in anemia.
That's a really important clinical point. Okay, this brings us to a common tool, the pulse oximeter. How does that little finger clip actually measure oxygen levels using hemoglobin?
It's quite ingenious, really. Pulse oximeters exploit the fact that oxygenated hemoglobin, HbO2, and deoxygenated hemoglobin, Hb, absorb red and infrared light differently.
Different colors, different absorption.
Right. The device shines both red and infrared light through a pulsating vascular bed, usually your fingertip or earlobe. It specifically looks at the light absorbed by the pulsatile component of blood flow.
Why pulsatile?
Because the pulse represents the fresh arterial blood surging through with each heartbeat. By measuring how much red and infrared light is absorbed by that arterial blood pulse, it can calculate the ratio of oxygenated hemoglobin to total hemoglobin, giving you the arterial oxygen saturation, or SpO2.
Fascinating. So it's cleverly isolating the arterial blood signal?
Exactly. It's a fantastic non-invasive tool. But there's a major pitfall, a really crucial clinical nugget to remember.
Okay.
Pulse oximeters cannot reliably detect carbon monoxide poisoning.
Why not?
Because carboxyhemoglobin, that's hemoglobin bound to carbon monoxide, HbCO, absorbs light very similarly to oxygenated hemoglobin, especially at the wavelengths used.
Oh, wow.
So a patient could have critical CO poisoning with their hemoglobin unable to carry or release oxygen effectively. But the pulse oximeter might show a reassuringly normal or even high saturation reading.
That's incredibly dangerous.
It can be.
Yeah.
It creates a false sense of security. You need a different test, a co-oximeter and a blood gas analysis, to actually measure HbCO levels.
Good to know. Okay, so we know hemoglobin carries oxygen and the curve shows how it releases it, but the body needs to be smart about where it releases more oxygen, right? How does it fine-tune this?
Absolutely. It's not just about the baseline PO2 drop. Metabolically active tissues, like your muscles when you're exercising, generate specific signals.
Signals like
Heat, increased carbon dioxide, CO2, and increased acidity, lower pH. These are the local cues that tell hemoglobin, hey, we need more oxygen right here, right now.
And how does hemoglobin respond?
These factors, higher temperature, higher CO2, lower pH, all shift that oxygen-hemoglobin dissociation curve to the right.
Shift to the right. What does that mean practically?
A right shift means that for any given PO2, hemoglobin has a lower affinity for oxygen. It holds on to it less tightly.
So it unloads oxygen more easily.
Exactly. It dumps oxygen more readily in those active tissues that are producing heat, CO2, and acid. It's a beautifully adaptive mechanism.
Let's break those down. Temperature first.
Sure. Increased temperature, like in active muscles, weakens the bond between oxygen and hemoglobin, shifting the curve right. So warmer tissues get more oxygen unloaded. Conversely, lower temperatures shift it left, increasing affinity, think hypothermia.
Okay. And then the big one, the Bohr effect. Christian Bohr noticed that acidosis shifts the curve right. How do pH and CO2 actually do that?
Right, the Bohr effect is really two related things happening. First, the pH effect. When blood flows through active tissues, it picks up CO2. Inside the red blood cell, CO2 reacts with water, sped up by carbonic anhydrase, to form carbonic acid, which then releases hydrogen ions, H+.
Making it more acidic, lowering the pH.
Correct. These hydrogen ions can bind to specific sites on the hemoglobin molecule, different from the oxygen binding site. This binding causes a conformational change that decreases hemoglobin's affinity for oxygen.
So the H+ pushes the oxygen off.
That's a good way to think of it. And conveniently, hemoglobin acts as a great buffer, soaking up many of these H+ ions, which helps limit the change in blood pH.
Okay, that's the pH part. What about CO2 itself?
CO2 itself can also directly bind hemoglobin, specifically to the amino groups on the globin chains, forming something called carbamino compounds or carbaminohemoglobin.
So CO2 binds directly, not just indirectly via pH.
Yes, and this direct binding of CO2 also caused a conformational change that reduces oxygen affinity, shifting the curve further to the right. That's the CO2 Bohr effect.
Wow. So active tissues create this perfect storm, lower pH and higher CO2 to ensure they wrench the oxygen off hemoglobin.
That's a brilliant physiological feedback loop. The waste product to CO2 and the consequence of activity acid directly enhance the delivery of the fuel.
Amazing. And there's one more player, right? 2,3-DPG.
Yes, 2,5,3-D-glycerate, or 2,3-DPG. This is a molecule produced during glycolysis, the way red blood cells generate energy since they lack mitochondria.
And how does it influence oxygen delivery?
2,3-DPG binds preferentially to the deoxygenated form of hemoglobin, the T state. It wedges itself into a central cavity between the beta chains.
Okay.
By binding there, it stabilizes the low-affinity T state, making it harder for a hemoglobin to transition back to the high-affinity R state.
So it makes it easier to stay deoxygenated, meaning it promotes oxygen release.
Exactly. It effectively shifts the dissociation curve to the right, just like increased temperature, CO2, and acidity do. It promotes oxygen unloading in the tissues.
And does the body adjust 2,3-DPG levels?
It does. In conditions of chronic hypoxia, like living at high altitude, or in chronic lung disease, or even severe anemia, the red blood cells increase their production of 2,5,3-DPG.
Why would it do that?
It's an adaptation. By increasing 2,3-DPG, the curve shifts further right, ensuring that even if oxygen loading in the lungs is slightly compromised, the tissues can still extract oxygen more effectively from the hemoglobin that is available. It prioritizes delivery.
That makes sense. Is this related to why fetal hemoglobin behaves differently? It has a higher oxygen affinity, right?
Exactly. Fetal hemoglobin, HbF, needs to pull oxygen from the mother's blood across the placenta. It achieves a higher oxygen affinity compared to adult hemoglobin, HbA.
How? Does it just inherently bind O2 better?
Not quite. The key difference lies in how it interacts with 2,4,3-DPG. HbF has gamma chains instead of the beta chains found in HbA. These gamma chains don't bind 2,4,3-DPG as strongly.
Ah. So less 2,3-DPG effect.
Right. Since less 2,3-DPG binds to HbF, it doesn't get stabilized in the low-affinity T state as much. Its curve is effectively shifted to the left compared to maternal HbA.
Allowing it to grab oxygen from the mother's hemoglobin in the placenta?
Precisely. It's a fantastic adaptation ensuring the fetus gets enough oxygen.
Okay, one more critical point about hemoglobin, carbon monoxide. We touched on the pulse oximeter issue, but why is CO poisoning itself so dangerous at the molecular level?
It's dangerous for two main reasons. First, as we mentioned with the pulse oximeter, CO binds to the exact same iron site on hemoglobin where oxygen binds.
Okay.
But its affinity for that site is about 200 times greater than oxygen's affinity.
200 times?
Yes. So even small amounts of CO in the air can effectively outcompete oxygen and tie up a significant fraction of hemoglobin, drastically reducing the blood's maximum oxygen carrying capacity.
So it steals the seats on the bus.
It steals the best seats, yes. But that's not even the worst part.
What's worse?
The second major reason it's so toxic is that when CO binds to one or more subunits of a hemoglobin molecule, it locks that molecule into the high-affinity R state.
The R state. But that loves oxygen.
Exactly. It shifts the dissociation curve for the remaining oxygen-binding sites dramatically to the left.
Ah, so the oxygen that is still bound can't get off.
Precisely. Hemoglobin holds on to the remaining oxygen much more tightly. So not only is the carrying capacity reduced, but the oxygen that is carried cannot be effectively released to the tissues where it's needed.
So the tissues starve even if there's some oxygen technically in the blood.
Yes. The blood might look bright cherry red because of the HbCO, but the tissues are profoundly hypoxic. It's a deadly combination.
Understood. Okay, so we've delivered the oxygen, dealing with all those complexities. Now, what about the return journey? How do we get rid of the carbon dioxide, the waste product?
Right. CO2 transport. Blood carries CO2 back to the lungs in a few different forms.
Yeah.
But the vast majority, something like 90% of the total CO2 in arterial blood, exists as bicarbonate ions, HCO3, dissolved in the plasma.
90% is bicarbonate. What about the rest?
Smaller amounts are simply dissolved CO2 gas in the plasma and inside red blood cells, and some is bound to proteins, mainly hemoglobin, as those carbamino compounds we mentioned earlier.
Okay, so bicarbonate is the main storage form. But what about the CO2 that's just picked up from the tissues? How is that initially handled?
That's where the proportions are a bit different for the change in CO2. Of the incremental CO2 that moves from tissues into the blood, in the systemic capillaries. About 10% stays as dissolved CO2, about 21% binds to hemoglobin to form carbaminohemoglobin, and the largest chunk, about 69%, is rapidly converted into bicarbonate.
And the red blood cell is key here again, right?
Oh, absolutely central. As CO2 diffuses from the tissues into the red blood cell, two crucial things happen almost simultaneously.
Okay, what are they?
First, the enzyme carbonic anhydrase, which is incredibly fast, grabs that CO2 and combines it with water to form carbonic acid, H2CO3.
Right, we mentioned that enzyme.
Yes, and this carbonic acid immediately dissociates into a hydrogen ion, H+, and a bicarbonate ion, HCO3.
Okay, so bicarbonate is made inside the red cell.
Correct. Second, to prevent bicarbonate from building up inside the cell and stopping the reaction, there's a transporter protein in the red blood cell membrane. It's called the chloride bicarbonate exchanger, or AE1.
What does it do?
It rapidly pumps the newly formed bicarbonate out of the red blood cell and into the plasma. In exchange, it brings a chloride ion, Cl, into the red blood cell from the plasma.
Ah, trading bicarbonate for chloride.
Exactly. This is the famous chloride shift, or sometimes called the hamburger shift.
Why is that shift so important? Why kick the bicarbonate out?
It serves a couple of purposes. It maintains the electrochemical gradient, and, crucially, it keeps the intracellular bicarbonate concentration low. This allows carbonic anhydrase to keep converting more CO2 as it enters the cell. It basically facilitates the continuous uptake and conversion of CO2.
So it keeps the CO2 processing factory running efficiently.
You got it. And remember, those hydrogen ions produced alongside bicarbonate?
Yeah, what happens to them?
They are largely buffered by hemoglobin, especially deoxygenated hemoglobin, which is conveniently becoming more available as oxygen is unloaded. Deoxyhemoglobin is a better buffer than oxyhemoglobin.
So the process of unloading oxygen actually helps buffer the acid produced from carrying CO2.
Perfectly coordinated. It minimizes the pH change within the red blood cell and the blood overall.
And I read that the red blood cells actually swell slightly as they go through the systemic capillaries because of this chloride shift.
That's right. As chloride ions enter, water follows osmotically, causing a slight increase in red cell volume in venous blood compared to arterial blood. A neat little consequence.
Okay, so just like oxygen, CO2 has its own dissociation curve, showing how much CO2 the blood carries at different pCO2 levels.
Yes, it does. And what's fascinating when you compare it to the O2 curve is that the CO2 dissociation curve is much steeper and much more linear over the physiological range.
Steeper and more linear, what does that mean?
It means that a relatively small change in pCO2 is needed to load or unload a significant amount of CO2. For instance, the pCO2 only increases from about 40 millimeter Hg in arterial blood to about 46 millimeter Hg in venous blood.
Just a 6 millimeter Hg difference.
Right. And that small difference is enough for the blood to pick up the required 4 mLDL of CO2 from the tissues. Compare that to the much larger PO2 drop, like 60 millimeter in Hg, needed to unload a similar volume of oxygen.
So CO2 transport is very sensitive to small pressure changes.
Extremely sensitive, which is efficient for removal. And this leads us to the Haldane effect, which you mentioned is kind of the flip side of the Bohr effect.
Right. How does that work?
The Haldane effect essentially states that deoxygenated blood can carry more CO2 than oxygenated blood can at any given pCO2.
Okay, why is that?
It comes back to what we just discussed. As hemoglobin unloads oxygen in the tissues, it becomes deoxygenated. And deoxyhemoglobin is better at two things related to CO2 transport. One, it's a better buffer for those hydrogen ions produced when CO2 becomes bicarbonate. Two, it's better at forming those carbamino compounds by binding CO2 directly.
Ah, so dumping oxygen makes hemoglobin better at picking up CO2 in both its main forms, indirectly as bicarbonate via buffering H+et, and directly as carbamino.
Exactly. So the very act of unloading oxygen in the tissues enhances the blood's ability to pick up CO2.
And the reverse happens in the lungs.
Precisely. As blood flows through the lungs and picks up oxygen, hemoglobin becomes oxygenated. Oxyhemoglobin is a poorer buffer and less able to form carbamino compounds. This reduced CO2 carrying capacity helps to push CO2 out of the blood and into the alveoli to be exhaled.
It's this beautiful reciprocal relationship. Bohr effect helps unload O2, where CO2 is high, and Haldane effect helps load CO2 where O2 is low.
Perfectly put. They work in concert to maximize gas exchange efficiency at both the tissues and the lungs.
So we've seen how O2 transport influences CO2 and vice versa. They're clearly in this constant, intricate dance. Is there a way to sort of visualize this interplay, bring it all together conceptually?
Yes, there is a way, though it's usually shown graphically. It's called the O2-CO2 diagram. Imagine plotting PO2 on the x-axis and PCO2 on the y-axis.
Okay, PO2 versus PCO2.
On this diagram, you can draw lines called isopleths that represent constant amounts of either O2 content or CO2 content in the blood.
What do these lines show us?
Well, the O2 content lines aren't straight vertical lines. They slope upwards to the right. This shows that as PCO2 increases, moving up the y-axis, you need a higher PO2 moving right on the x-axis to maintain the same amount of O2 in the blood.
Ah, that visualizes the Bohr effect. Higher CO2 means lower O2 content at the same PO2.
Exactly. And similarly, the CO2 content lines aren't straight horizontal lines. They slope downwards to the right. This shows that as PO2 increases, moving right on the x-axis, you need a higher PCO2 moving up the y-axis to carry the same amount of CO2.
And that's the Haldane effect. Higher O2 means lower CO2 content at the same PCO2.
Precisely. The diagram beautifully integrates both effects and shows how the loading or unloading of one gas directly influences the transport of the other. It paints a complete picture of their interdependence.
What an incredible journey through the respiratory gases in our blood. I mean, from the complex structure of hemoglobin, that amazing cooperativity, to the brilliant dance of the Bohr and Haldane effects. It's just clear our bodies are master engineers.
They really are. And understanding these mechanisms isn't just, academic exercise. It's absolutely fundamental to diagnosing and treating a huge range of respiratory and circulatory conditions.
Right.
Those subtle shifts in the curves, the way gases are transported, they tell clinicians so much about what's happening at the tissue level. It guides really crucial decisions about oxygen therapy, ventilation, and more.
And just think about it. The very act of your tissues using oxygen creates the perfect local conditions, heat, acid, CO2, for your blood to release more oxygen right there, and simultaneously helps the blood pick up the CO2 waste.
It's an incredibly elegant, self-regulating system, constantly adapting moment by moment to your body's metabolic needs.
And the sheer speed and efficiency with which your red blood cells manage all this converting CO2, shifting ions, changing hemoglobin's affinity, it's truly one of the marvels of physiology.
It really is. It underscores how these cellular and molecular level details have profound impacts on your entire body's function, from taking a simple breath to running a marathon.
Well, we hope this deep dive has helped clarify these absolutely essential concepts for you. Keep exploring, keep asking questions, and keep learning.
You've definitely got this.